Thank you, Dr. Ross, for the opportunity. But really what my objective tonight is to give you really a conversational understanding of nitric oxide. It's probably one of the most complicated molecules and enzymatic productions of any given molecule in the human body. It has a half-life of less than 1 second, and so to be able to recapitulate nitric oxide-based signaling in physiology has been a great challenge. In fact, big pharmaceutical companies have worked on this for probably at least 30 years, hundreds of millions, if not billions of dollars now in terms of research and development. And so what I want to do tonight is share with you kind of my 20-year journey in basic science and to share with you some very simple observations that we had from basic science and how that made us change and pivot our understanding of how the body controls and regulates nitric oxide production. And so ultimately what I want to leave you here with is kind of some clinical pearls on how you can hopefully begin to recognize how you can apply nitric oxide-based therapies into patients that you probably otherwise wouldn't consider as a candidate for nitric oxide. And so as Warren said, I was at University of Texas Medical School for a number of years. I'm now at Baylor College of Medicine there in the Texas Medical Center. But I thought I could spend probably 15 minutes or 10 or 15 minutes on these three, really just giving you kind of some basic understandings around nitric oxide biochemistry and physiology. From a clinical perspective, how to recognize a nitric oxide deficient patient, how to test for this, what are the clinical consequences, and then at the end I'm going to wrap this 17 years of basic science into the discoveries we've made and how we can translate that into clinical medicine. And then I'll show you some case reports of how you can apply this, even in some rare genetic disorders, and really affect the outcome of some pediatric patients that otherwise had met the end of their clinical care. I'm required to disclose to you that I have both a financial and research conflict of interest. I'm the founder and shareholder of HumanN, a company we formed out of the University of Texas Medical School in Houston. They own all my patents. I have 8 issued U.S. and international patents, and then we license those patents back from UT. I'm a shareholder and advisor for Sage Pharmaceutical, and I'll show you, I won't spend a lot of time on that, but we've developed some natural product inhibitors of a key nitric oxide metabolizing enzymes. And then I receive royalties on my patents from University of Texas Health Science Center, Houston. So the first question I ask you is, who would be a candidate for nitric oxide? And I think at the end of this seems like an exhaustive list, but at the end of this, hopefully I'll present the science that will convince you that this isn't really unreasonable in terms of who would be a nitric oxide candidate. Certainly anybody who's aging, I'll show you this age-related decline in nitric oxide production. Anyone who's sick, so if a patient's coming to a physician's office, obviously they're sick. We're not at a stage in medicine now where people are proactive. They come to a physician to figure out how not to get sick. I hope that's where we will be in the future, but that's not the way medicine's practiced today. Certainly anybody over the age of 40, because by the time we reach 40, we only have 50% of the nitric oxide we have when we were younger. Circulatory issues, diabetics. I'll show you some key basic science discoveries that will hopefully convince you that loss of nitric oxide is causal for type 2 insulin-resistant diabetes. You restore nitric oxide in diabetic patients, insulin signaling, glucose uptake, all the vascular complications of diabetics can be prevented. People with low energy, I don't have time to go into it tonight, but nitric oxide controls and regulates mitochondrial ATP production and mitochondrial biogenesis. People's sexual dysfunction, erectile dysfunction is really the first sign and symptom of nitric oxide deficiency in both men and women. People on antacids, proton pump inhibitors, or I think my goal is to get this in the hands of people who aren't sick, but yes, want to prevent from getting sick. And I think if we take it back to this very simple concept of vascular biology, that a man is as old as his arteries, then this begs this fundamental question of how do we prevent arterial aging? But we know through even post-mortem analysis of young patients from motor vehicle accidents that when you analyze their coronary arteries or even other vasculature, that there's fatty streaks in the lining of the coronaries. But as you age, you get this progression of carotid intima media thickness, or not just in the carotids, but intima media thickness, where you get plaque develop. Over time, this plaque becomes unstable, it ruptures in heart attack or stroke, and typically that's the end of the story. We now have these diagnostic biomarkers that can tell us at each stage of this progression of vascular disease. So the earliest stage of vascular disease is really this microvascular inflammation, where you get an increase in a biomarker called high-sensitivity C-reactive protein. you begin to spill some protein in the urine. Later stages, you get upregulation of myeloperoxidase, changes in lipoprotein particle size, number, and density. And then diagnostic for acute MI is creatine kinase or troponin T. So our objective was to be able to try to understand where does nitric oxide fit in the spectrum of vascular disease. And we've been able to mirror that now to look at endothelial function or endothelial dysfunction and this onset and progression of vascular disease. And so you can basically mirror this whole progression of disease about how well this patient makes nitric oxide. So our strategy is let's start looking at functional changes in endothelium or endothelial function before there are any structural changes and changes in these inflammatory biomarkers or diagnostic biomarkers in vascular disease. And so that takes really a change in paradigm and new treatment and diagnostic modalities to understand what nitric oxide status is in individual patients, hopefully before they get sick. This is the molecule, I think, obviously with my bias, probably one of the most important molecules, but it's very simple. It's one nitrogen, one oxygen, has a free electron, so it's a free radical. And most people were thought that these free radicals were toxic, damaging molecules, and oxygen radicals are because they'll scavenge other electrons. But because of the polarity of this nitrogen and oxygen bond, NO actually acts as an electron donor. So any electron donating species has a potent anti-inflammatory, antioxidant properties, and it can scavenge a lot of these toxic oxygen radicals like hydroxyl or superoxide anion. As I mentioned, it has a half-life of less than 1 second. This is just kind of it's not nitrous oxide for the dentist in the room. This is completely different. They sound the same, but they're completely different in terms of their physical chemistry. There are now over 140-something thousand papers published in the medical literature, and so I'm still surprised that people don't know about nitric oxide. Physicians, clinicians can no longer ignore the science. This is a Nobel Prize was awarded in 1998. The first discoveries of nitric oxide happened in the late 70s, early 80s. And so it's now recognized as a ubiquitous cell signaling molecule that's involved in every biological and cellular function in the human body, from regulation of blood pressure and perfusion to every tissue and cell in the body, to the non-adrenergic, non-cholinergic mediator in the periphery. It's a neurotransmitter in the central nervous system, controls cell cycle, and the main molecule responsible for erectile function in both men and women. And it's actually how our immune system fights off invading pathogens, through upregulation of nitric oxide production. And as Dr. Ross mentioned, the Nobel Prize in Physiology or Medicine was awarded in 1998 to these 3 U.S. scientists. And it was Fred Murat who discovered that drugs like nitroglycerin, organic nitrates, that were used for probably 150 years prior to the 70s for the treatment of acute angina, worked because they were metabolized into nitric oxide, they dilated coronary arteries, and could get complete symptomatic relief of the ischemic pain from angina. In 1980, 3 years later, Bob Furchgott discovered that our endothelial cells makes a substance that dilated blood vessels. And he called this endothelium-derived relaxing factor, or EDRF. So clearly knowing that what it did, but he had no idea of what it was. And then Lou Ignarro discovered 7 years later that EDRF is nitric oxide. And so then the pieces of the puzzle were put together, because if you stimulate endogenous nitric oxide production, dilates blood vessels, if you give an exogenous NO donor, it can still stimulate smooth muscle relaxation and vasodilation. And I've been fortunate. I've worked with Fred. Fred recruited me to join the faculty of UT Medical School in Houston back in 2005. So a lot of the data I'm going to show you was with Fred as my department chair. And then Lou has written the foreword to several of my textbooks. And this was the front page of the New York Times the day the Nobel Prize was awarded in 1998. The discovery of NO and its function is one of the most important in the history of cardiovascular medicine. That was by Val Fuster, who's then president of the American Heart Association. And I show you this because for years we've thought this in the NO field, but it wasn't until recently that Jonathan Stamler at Case Western revealed unequivocally that nitric oxide is critical for oxygen delivery into the periphery. So there's a beta cysteine residue on a cysteine residue on the beta chain of hemoglobin that NO binds to. And so if you remember back from basic biochemistry that when hemoglobin goes from the arterial to venous transit in capillaries, it goes from this R to T transition. And during that allosteric regulation, it offloads oxygen and picks up CO2. But without nitric oxide bound It cannot offload oxygen. So if you think of this in terms of critically ill patients that are getting 100% oxygen through a nasal cannula, they may have 98% O2 sats, but they're peripherally hypoxic because they've got bound oxygen, but they can't offload the oxygen. So we have to start thinking about restoring the nitric oxide signaling aspects of not just vasodilation, but hemoglobin delivery of oxygen to the periphery. And these patients can't get better until you restore their nitric oxide. And obviously, if you can't deliver oxygen to tissues and cells of the body, you slowly begin to die. And that's what we call aging. So how do we fix this fundamental problem? It's a very simple question I asked many years ago. And so to get to the answer to this question, you have to understand the basic cellular biology or vascular biology of how nitric oxide is produced. So if you can appreciate, this is a single layer of endothelial cells that line all blood vessels and lymphatics throughout the body. This would be the lumen of the blood vessel. And this is the smooth muscle that surrounds all blood vessels. Then when smooth muscles contract, you get a constriction of blood vessels. When smooth muscle relaxes, you get dilation. So when we begin to exercise, this induces shear stress. That's why exercise is medicine. And when you have a functional endothelium, that tells this enzyme to make nitric oxide. And the first pathway to be discovered was the oxidation of arginine to nitric oxide. And so once nitric oxide is produced, it's a gas, so part of it diffuses into the lumen of the blood vessel where it interacts with platelets and monocytes and neutrophils, prevents them from sticking to the lining of the blood vessels. But part of it diffuses into the underlying smooth muscle where it activates an enzyme called guanylyl cyclase. And this is the enzyme that Dr. Muir had worked on for a number of years that converts GTP, or guanosine triphosphate, into cyclic GMP. And then cyclic GMP is the second messenger that elicits this calcium-dependent mechanism that causes smooth muscle relaxation. This is also the mechanism of action of PD5 inhibitors or erectile dysfunction drugs. So there's an enzyme called phosphodiesterase that degrades cyclic GMP into GMP and inorganic phosphate. And so if you can prevent the breakdown of cyclic GMP, you can potentiate vasodilation and improve erections in men with ED. And so these drugs are obviously very successful, but they only work in 50% of the men in which they're prescribed. So the etiology of erectile dysfunction isn't an overactive phosphodiesterase enzyme, it's insufficient nitric oxide production in the endothelium. Because if you can't make nitric oxide to lead to an accumulation of cyclic GMP, PD5 inhibitors can't work. So now we've got clinical data. We've just recently finished enrollment of a study where we're taking people who have been on PD5 inhibitors for 90 days that have suboptimal erections or non-responders and restore their nitric oxide production and now make them responders to PD5 therapy. And this is a busy slide, but the point here is to convince you or tell you that the literature tells us that loss of nitric oxide is really the first event in the onset and progression of vascular disease. And as we age, our endothelial function becomes more compromised. In fact, about 10 to 12% loss of endothelial function per decade. And so by the time we're 40 or 50 years old, we only have about 50% of the nitric oxide we had when we were younger. And so I don't think it's coincident that that's the time you start to feel the effects of aging. You don't perform as well as you did in the boardroom, the athletic field, or even the bedroom. In fact, 50% of the men over the age of 40 have some signs of erectile dysfunction. Again, leading to this causal event of loss of nitric oxide. So when you lose the ability to make nitric oxide, how do these patients present? Obviously, their blood pressure increases. They have advanced atherosclerotic disease. Platelets have their own nitric oxide synthase enzyme that when it becomes disrupted, they develop coagulopathies. Alzheimer's or vascular dementia, when you lose the ability to perfuse certain regions of the brain on demand, you lose function. ED, PAD, immune dysfunction, uncontrolled cell proliferation, and even cancers. So again, it begs the basic questions of how do we control and regulate nitric oxide production? Because if all these diseases and symptoms that are age-related revolve around the loss of production of nitric oxide, if you fix that, then you can fix a lot of the downstream effects. And the take-home message here is it's damn complicated. And so people have spent their entire academic careers studying this single molecule or this enzyme called nitric oxide synthase. And so people thought for years, well, I'll just give arginine and generate nitric oxide. But there's an enzyme called nitric oxide synthase that through a 5-electron oxidation, converts this guanidino nitrogen into nitric oxide. And it's the dysfunction of this enzyme that occurs with age. And so the other important biochemical component here is that the Michaelis constant of nitric oxide synthase, which is the concentration of arginine that's needed to theoretically saturate 50% of the enzyme, is only 5 micromolar. And in the sickest of the sick patients, their plasma levels and intracellular concentrations of arginine are 100 to 200 micromolar. So more than 20 to 40 times higher than what's needed to saturate the enzyme. So this tells us that L-arginine is not the rate-limiting step. L-arginine deficiency is not the rate-limiting step in nitric oxide production. And it's the only human enzyme known to require 8 different cofactors and substrates. And really the rate-limiting step is oxidation of tetrahydrobiopterin, which uncouples this enzyme. And when you uncouple the enzyme, it generates superoxide instead of nitric oxide. So giving arginine to a patient with endothelial dysfunction can actually make things worse instead of making them better. We've discovered that we can extend the biological half-life of nitric oxide from 1 second out to tens of minutes or hours because it can bind to the cysteine residue of glutathione and be transported as an S-nitrosoglutathione. It's oxidized to nitrite, nitrate, and I'll show you how the oral bacteria are responsible for reconverting these oxidative metabolites into bioactive nitric oxide that can then affect systemic blood pressure. This is a 2011 Nature Medicine paper we published, and I'll bring this back at the end of this study from these urea cycle patients. But this was a fundamental discovery that we've discovered a new protein complex, that the regulation of nitric oxide production is part of the urea cycle, and that the cell doesn't use extracellular L-arginine to make nitric oxide. It uses arginine that comes from citrulline through the partial urea cycle. And so that arginine goes to make nitric oxide, you get citrulline as a byproduct, and then you've got this intramolecular shuttling of these substrates that's really spatially and temporally controlled of nitric oxide production. And if you don't have one of these enzymes, which is an inborn error metabolism, these patients become nitric oxide deficient, and I'll show you at the end of the talk of how we're able to fix a very rare genetic disorder simply by providing nitric oxide. So as Warren said, I'm from Texas. I ask very simple questions. We're not very complicated people in the South. And so the very basic question we asked years ago was, if we enhance nitric oxide production in the heart, could we prevent injury from heart attack? And then if we could protect the heart, could then we trace this phenotype biochemically and develop some biomarkers that had prognostic or diagnostic utility? So we used transgenic mice in this, and this was a study in collaboration with David Leffer, that you can overexpress the nitric oxide synthase enzyme only in the heart, in the cardiac myocytes. And this enzyme is active because it's spitting out a lot of nitric oxide that's oxidized to nitrite, nitrate, and these NO-modified proteins. And if you subject these mice to myocardial ischemia reperfusion injury, so you occlude the LAD for 45 minutes, then you reperfuse for 24 hours, there's no injury to the heart. So these mice are pumping out tons of nitric oxide. They have an acute MI, and there's no infarct in the heart. There's no necrosis. The echocardiographic analysis shows that there's no functional impairment of these hearts that have been no-flow ischemia for 45 minutes. And so we begin to look at what's happening in other tissue. So the heart's protected, but what's happening in other tissue? So we took the liver of these mice and found that the liver biochemistry was modified. So understanding that nitric oxide couldn't survive transit from the heart to the liver, there had to be something else happening. And it was a clean transfection because there wasn't overexpression of the enzyme. And so we go, what's the meaning of this? And so if we occlude the blood flow to the liver for 1 hour and then reperfuse for 5 hours, you can completely protect the liver from damage as measured by these liver enzymes. And so these data took us probably 3 years to get published because no one believed the data. There was actually a hormone function or an endocrine function of nitric oxide, that nitric oxide produced in the heart could protect the liver from injury. And so this PNAS paper from 2008, I think, was the first demonstration that there was an endocrine effect of nitric oxide. And so this was my aha moment, because really if we wanted to affect distal organ protection, all we had to do was generate nitric oxide in a single biological compartment. And then moving to another kind of therapeutic strategy, that is 50% of the nitric oxide, total body nitric oxide, was determined by foods and the presence or absence of oral bacteria, then this offered a new therapeutic strategy to restore nitric oxide. So again, we did some very simple experiments. We did a dietary intervention in mice. We excluded nitrite and nitrate from their diets. So nitrate comes primarily from green leafy vegetables. The standard rodent chows have some in there, but if we delete that from the diet, we can affect the the biochemistry. We can downregulate these NO metabolites. If we give it back in the drinking water, we can actually restore back to normal control mice. And if we do in vivo LAD ligation in these mice, you can see without sufficient nitrite and nitrate from the diet, there's about 30% more infarct in the heart. 50% of these mice don't survive out to 24 hours. So increased mortality, increased infarct. If we give it back in the drinking water, 100% survive out to 24 hours, and there's less infarct in the heart. So this gave us indication that we could affect the outcome from acute MI simply by a dietary intervention. And there was evidence being presented in the literature that if you gave beetroot juice, there was a lot of athletes after the 2012 Olympic Games in London that were drinking beetroot juice because it enhanced their performance. And you could actually reduce blood pressure and affect platelet activity simply by drinking beetroot juice. And so the problem was, and we began to get into that, but there were so-called non-responders. We thought it was these genetic single nucleotide polymorphisms. And so we began to look at different testing modalities of different SNPs. And this is where basic science really leads to a disservice to clinicians, because for 17 years I've focused simply on this reaction. And that happens in basic science. We forget that everything is connected to gears and wheels. So if this pathway is disrupted, everything backs up biochemically. And similarly, if an MTHFR SNP is a problem, then it affects everything downstream and upstream. And so the reason I show you this is because the methyl tetrahydrofolate reductase enzyme, and these are very common SNPs in patients, is that by definition they become nitric oxide deficient because they've lost the ability to reduce BH4 to BH2 to BH4, which is the rate-limiting step in nitric oxide production.
Excuse me, is that true if they're heterozygous versus homozygous?
Well, heterozygous, they just have 50% of the enzymatic activity. So they still are nitric oxide deficient. But especially the homozygous MTHFR patients, by definition, become nitric oxide deficient. And I want to mention a little bit about diabetes because it's an epidemic. It's probably the number one disease that's modifiable by diet and lifestyle, but compliance is an issue. And so we've known for a number of years that once diabetics get diabetes, it downregulates nitric oxide production. And typically diabetics don't die from diabetes, they die from the vascular complications of diabetes. And all this is because it leads to a dysregulation of nitric oxide production. But several decades ago, they knocked out the eNOS enzyme in mice, and the first phenotype these mice develop is type 2 insulin-resistant diabetes. So suggesting that loss of nitric oxide production may be causal for type 2 diabetes. And then once you get diabetes, it further suppresses nitric oxide production, and that's why these patients are so complicated in terms of the vascular effects of diabetes. And this is a very interesting observational study published by the Karolinska Institute back in 2010. So they took these eNOS-deficient mice and gave them inorganic nitrate in their drinking water. So similar to what you would get from eating some plate of spinach or green leafy vegetables. As you can see here, they gain less body weight even though they're drinking the same amount of water, eating the same amount of food, about a 20% reduction in triglycerides, about a 40% reduction in visceral fat deposition. Glucose tolerance improved. Hemoglobin A1C was reduced by 50%. Better blood pressure management. And so overcoming a genetic deletion of an enzyme simply by giving a component or a molecule that can reduce back to nitric oxide could completely overcome this genetic deletion, phenotypically in terms of diabetes. And so that was an interesting observation, but it didn't tell us how this was happening. So we began to interrogate the insulin signaling pathway because the literature told us that once insulin binds to the insulin receptor, it activates this series of kinases, primarily PI3, AKT kinase, that phosphorylate the eNOS enzyme, or even AMP kinase, which is where metformin acts, activates this enzyme. All these converge on the eNOS enzyme, and it turns this enzyme on if the enzyme is functional. But we know in diabetics that this enzyme can't make nitric oxide. And so that's where the literature stopped. But we knew the terminal step in glucose uptake was GLUT4 translocation. So there was this disconnect between what happens with nitric oxide production and GLUT4 translocation. And so we basically solved this riddle by engineering some cells that had green fluorescent protein co-localized with GLUT4. So the lighting may not allow you to appreciate this, but we can actually see in real time GLUT4 translocate to the membrane, increase glucose binding and glucose uptake, and improve insulin signaling. And we published this several years ago, but mechanistically we've worked this out because there's 2 cysteine residues on GLUT4 that you can post-translationally modify through nitric oxide. And when you nitrosylate the cysteine residue, that's necessary and sufficient for GLUT4 translocation. So if diabetics, which are nitric oxide deficient, there's a disruption in insulin signaling. If you restore nitric oxide production in diabetics, you can post-translationally modify this GLUT4 protein, translocate GLUT4, increase glucose uptake, and improve triglycerides, fasting glucose, and hemoglobin A1C levels in diabetics, and improve endothelial function in diabetics. Now I'm going to show you some unpublished data because to me this is probably the most interesting thing and will, I think, change the whole nitric oxide field. But I want to present to you that hypertension may not be a vascular disease or a symptom of vascular dysfunction, but maybe a symptom of oral dysbiosis. And if that's the case, then obviously this offers a new treatment modality. And we know this because this makes sense in terms of nitrate reduction to nitrite. Humans don't have a nitrate reductase enzyme. So we're 100% dependent upon oral bacteria. And then nitrite reduction to nitric oxide is a very inefficient process. We published this in Nature several years ago, or over a decade ago. And so the natural product chemistry we've discovered can actually take this 0.01% reduction efficacy into 90%. So we can generate nitric oxide along the entire physiological oxygen gradient, whether it's on the arterial side or the venous side, on demand that's unaffected by oxygen. And this isn't a new concept. It's let food be thy medicine, medicine be thy food, the foundation of modern medicine. And so this begs the question of how much nitrate do we need? How much spinach do you need to eat if you want to affect blood pressure or reduce your blood pressure? And the literature tells us that you need at least 300 to 400 milligrams as a bolus to see any changes in blood pressure. But we know that the U.S. population of the so-called Western diet, you're only getting about 150 milligrams, and that's over 2 to 3 meals per day. So we become a nitrate-deficient diet, the population. And so the next question we asked was, well, how do we change the dietary guidelines? If we want to let food and diet and nutrition be our first line of defense against disease prevention or regulation of blood pressure, then how much broccoli do you need to eat? How much celery do you need to eat? Or how much kale or spinach? And so we did this study several years ago, and we went around to 5 U.S. cities. We went to Chicago, Dallas, Los Angeles, New York, and Raleigh, the 5 corners of the U.S. And we went to a single retail grocer and bought broccoli, cabbage, celery, lettuce, and spinach off the shelf, brought it back to the lab, and analyzed it. And so the purpose was to say, okay, how much nitrate are you going to get from these vegetables? And so the answer was, if you live in Dallas, you can eat about 4 stalks of celery and get enough nitrate to lower blood pressure. If you live in New York, you need about 40 sticks of celery to affect blood pressure. The same thing happens with broccoli, cabbage. I mean, there's as much as a 50-fold difference in these. So there's no way in hell we can make dietary recommendations to affect blood pressure because there's regional differences on how crops are grown, this whole field of agronomy, what the nitrate concentration is. And these are conventionally grown vegetables. Organically grown vegetables to which there's no standardization of fertilizers, the variation's even higher. In fact, they have less nitrate than conventionally grown. They have less. They're free of pesticides and herbicides, but they have less nitrate. And so that got us asking the question, well, nitrate's inert in humans, so how do the oral bacteria play a role in responders to spinach? And you've probably all heard of cases where people have sudden cardiac arrest. Some people die from sudden cardiac arrest. Other people survive it, and their cardiac function or ejection fraction is somewhat normal after that. So I would argue that you got to ask these patients what they had as their last meal. Did they eat a plate of spinach 3 hours before? And so I'll take you through the role of the oral microbiome and how this plays a role. This is a study where you took normal-tensive, healthy individuals, give them chlorhexidine mouthwash for 7 days, and their blood pressure increases about 2 to 3 millimeters of mercury, showing for the first time that affecting the oral microbiome affects systemic blood pressure. This shouldn't be surprising because we humans only generate about 23,000 gene products. The bacteria that live in and on our body contribute about 3 million gene products. And this is true symbiosis. These bacteria are there to do things that we as humans haven't evolved to do. And this is a very simplistic scheme. And so soil bacteria, and this is the basis of fertilization, can take ammonia or elemental nitrogen and fix this nitrogen into usable forms by food or by plants. And in the plants that we eat, we get a source of this nitrogen. But the bacteria that live in the crypts of our tongue are denitrifying bacteria. So they take nitrate and reduce it down to nitrite, nitric oxide, and some can reduce it all the way down to elemental nitrogen. So we did some studies at the Texas Medical Center where we just took tongue scrapings of people walking around the Texas Medical Center to try to figure out what bugs do they have growing in their mouth and what does this have to do with nitric oxide production. In this early study, we found that different patients had different communities that, based on their nitrate reductase activity, begin to cluster. And so we published this several years ago, but the people that could reduce nitrate the best had different bacterial communities in their mouth than those who couldn't. So then we asked the question, well, how can we intentionally disrupt these oral communities and allow the certain bacteria to repopulate, and does this have any effect on blood pressure? And this is unpublished data. We've got this in Nature Medicine that I hope will be accepted. It's under review now. But this completely changes the whole thought on how we treat hypertension. And this was the design of the study. We had, I think, 34 patients. These are normal-tensive, healthy individuals, primarily dental school. This study was done at UT Dental School in Houston. A lot of dental students, dental hygienist students, some faculty. But we get blood pressure, we get their oral microbiome, we give them chlorhexidine twice a day for 7 days, monitor their blood pressure over these 7 days, then allow a 7-day off period. What bacteria recolonize and how does this affect blood pressure? Again, very simple, but this was a booger of a study. But what you see here is some people's blood pressure went up with chlorhexidine treatment, some blood pressure went down, some people there was no effect. So we begin to interrogate the so-called responders and the outliers. This was one of our outliers. This was a 21-year-old dental student, and 7 days of chlorhexidine treatment, his systolic blood pressure went up 26 millimeters of mercury. So we made this kid clinically hypertensive simply by giving him a chlorhexidine. And so we begin to do tongue scrapings every day, and you can see here that as the blood pressure went up, certain bacteria disappeared. As blood pressure normalized, these bacteria begin to repopulate. And then we actually looked at the metabonomic effects of this and looked at the expression of certain enzymes of these bacteria. And then we asked the question, we modified the IRB to get some more insight into these patients and found what was their oral hygiene. And so how many people brush their teeth, actually scrape their tongue, if they scrape their tongue, how often, how many use mouthwash. So all these things are contributing to this variability in the effect of not just steady state blood pressure, but the effects of blood pressure from chlorhexidine. And so when we ran the statistics on this, we found that the change in blood pressure from chlorhexidine correlated quite nicely, highly statistically significant, with tongue hygiene. So the people who cleaned their tongue had lower steady-state blood pressures, but if you give them chlorhexidine, their increase in blood pressure was most dramatic. And then we started looking at the genetic expression of these denitrifying enzymes in the bacteria that were growing in the mouth. And this is the money slide here. We can predict steady-state blood pressure by the presence or absence of 4 bacteria. And this is independent of treatment of chlorhexidine, but really your steady-state blood pressure can be predicted by the oral microbiome in these 4 bacteria. Now we've got data in older resistant hypertensive patients that are unresponsive to standard antihypertensive therapy. And if we look at these bacteria, they're completely absent. So the next question is, I don't like to provide problems without providing some solutions, or state problems without providing solutions. So how do you overcome this? So obviously this is multifactorial. Number one, we don't get enough nitrate in our diet to be reduced to nitric oxide. And because everybody's a germaphobe and wants to use antiseptic mouthwash, overuse antibiotics, we're killing a lot of these good bugs. And so I think it can be argued that we are a nitrite deficient society. And is this causal for disease? a concept that Linus Pauling put forth several years ago. Hypertension is a huge problem. 2 out of 3 Americans have hypertension or prehypertension. That's about 200 to 300 million people that we think we can affect. 50% of those on antihypertensive medications are unmanaged. And the SPRINT trial, probably many of you are familiar with it, was published last year, shows that if you can reduce blood pressure to just 120/80, that you can reduce all-cause mortality. So we have to do a better job of managing blood pressure. And so the thing that understanding how this process works, we thought, well, can nitrite overcome the variability in the oral microbiome if we're nitrate deficient? There's certain acute toxicities of this molecule. It's mainly methemoglobinemia and hypotension. And so if you consume about 1.85 or 2 grams of nitrite, you'll get methemoglobinemia, you get low blood pressure, you get end-organ failure, and you'll die. Bad things happen. But you can never consume enough vegetables or hot dogs or cured and processed meats to ever get a fatal dose of nitrite. The long-term toxicity is really this propensity to form N-nitrosamines, which are carcinogenic. But we know that there's ways to mitigate this chemistry. And so our concept has always been, if you're deficient in something, let's give it back. Very similar to vitamin D. If patients are deficient in vitamin D for whatever reason, you give it back to titrate up their levels to normal. And that's exactly what our philosophy is on nitrite, nitrate. We know how much of these anions the human body makes. If you're deficient, let's just give it back. Not more, not less, but give back and recapitulate normal human physiology. And so I'll take you through the clinical trials now, because this is really the culmination of about 15 years of work that we productized this technology, and this is the basis of the 8 issued patents at UT Medical School in Houston. And so we developed this technology called Neo 40. It's an orally disintegrating tablet. I see you've got some on your table. But this was a very deliberate design, that we wanted to have a specific resident time in the mouth that activated by your saliva to generate a certain amount of nitric oxide over a certain period of time, and then also fix the underlying problem of endothelial dysfunction. And so this is published clinical trials in the peer-reviewed literature. But as you put the lozenge in your mouth, it generates nitric oxide gas, about 25 to 30 parts per million. So those of you that are perhaps familiar with pediatrics, in premature babies born with premature hypertension, they give them 25 to 30 parts per million NO gas to reduce pressures. Same amount they give in the adult cath lab for pulmonary hypertension. The good thing about this is the pressure, the effects last much longer than the gas, and I'll show you those data. And so going back to that first slide of a man is as old as their arteries, then this begs the question of what causes arteries to age. And there are really 3 finite responses to the infinite number of insults that our body gives, whether it's glyphosate ingestion from eating foods, whether it's genetic mutations, or whether it's just poor diet and lifestyle. Our body responds, our vasculature responds with inflammation, immune dysfunction, and oxidative stress. And so there are a number of these, and I'll take you through these, but increased oxidative stress is characterized by myeloperoxidase activity, changes in oxidized LDL, but really triglycerides are an indirect measure of oxidative stress. It's apparent glucose metabolism. And so this shows that 30 days, taking 1 lozenge twice a day, we could reduce triglycerides by about 27%, highly statistically significant effect. In fact, that's one of the bases for the patents I have, is a method of reducing triglycerides. So oxidative stress, we're seeing reductions in myeloperoxidase activity, all of these oxidative stress markers, F2 isoprostanes in the urine. The other, and I think probably one of the most important aspects of vascular inflammation, is this immune dysfunction that occurs when monocytes and neutrophils start sticking to the lining of the blood vessels. They migrate through, they become modified LDL, foam cells develop plaques become unstable, they rupture, and that's the end. But it's also known that if you can prevent this earliest event of monocytes and neutrophils sticking to the lining of the blood vessels, you can prevent this whole cycle of vascular disease. And so this is real-time intravital microscopy of vascular inflammation. And so this is an inflammatory diet and the effects on vascular inflammation. So you can see the monocytes and neutrophils, they're sticking here. There's a little bit of traffic jam, a little bit of blood flow going through here. But this is a heightened state of vascular inflammation. And actually some monocytes have already migrated through. So this is a paper we published back in 2009, and we can assess an inflammatory score. if we give that same atherogenic diet, and this is a 6-week feeding study, but we give a source of nitric oxide to restore endothelial production of NO, you can see that the blood vessel is actually more dilated. The monocytes and neutrophils home along the endothelium, looking for sites of injury, looking to do their job, but they don't stick. So you can completely suppress the microvascular inflammatory response to a poor diet by giving nitric oxide. So I think we can check off the immune dysfunction aspect of that. And then inflammation, again, one of my patents is on a method of reducing C-reactive protein by 37% in 30 days. So I think we've demonstrated, at least in these 6 published clinical trials in humans, that we can inhibit the causes of arterial aging simply by restoring nitric oxide production. I'll show you the blood pressure effects of this, and I think it's worth mentioning that there are about 400 risk factors for cardiovascular disease. Most of all these can be modified by or corrected by modifying nitric oxide. Hypertension is the number 1 risk factor for the development of the number 1 killer of men and women worldwide. And so I'll show you the results of this study. This is a double-blind, placebo-controlled crossover study, the gold standard in clinical trial design. But we looked at, these are naive hypertensive patients, get baseline data, look at the ultrasound of the carotid for 10 minutes, get systemic blood pressure after 20 and 60 minutes, pulse wave analysis after 30 minutes, and then 4 hours later, long after the lozenge is dissolved, what happens to the enzyme in the endothelium that makes nitric oxide? Because if we really want to improve patients' health, we've got to fix the underlying endothelial dysfunction. 3-week washout and then cross them over. And this is the data published several years ago, but you can reduce systolic and diastolic pressures by about 4 to 5 millimeters of mercury within 20 minutes. 60 minutes, there's a further statistically significant reduction. And then 4 hours later, you see about a 15% improvement in endothelial function. So I think this demonstrates that we've done what we set out to do. Well, number 1 is if the patient can't make nitric oxide, we make it for them. And then number 2, we fix the underlying endothelial dysfunction, or recouple the nitric oxide synthase enzyme in the endothelium. Interestingly enough, the higher the pressure, the bigger the effect. If you have normal or low blood pressure, we're not further reducing your pressures, which is a very important safety consideration. This is the left common carotid. You can see here, maybe with the lighting you can't see, but baseline we can get this tonometer and get internal luminal diameter of about 0.52 centimeters. 10 minutes after the lozenge, the blood vessels dilated to 0.6 centimeters. And so this is a 13% increase in blood vessel diameter. If you apply simple laws of hemodynamics, that translates into a 34% increase in blood flow. And that's perfusing the brain. We see improvements in vascular dementia patients and in cognition, but this is just the carotid, but this also occurs in resistance arterioles, in feeding the skeletal muscle, feeding the sex organs, feeding the coronary arteries. This is the change in arterial compliance. Not only are we affecting systemic blood pressure, but central aortic pressure decreases, total vascular resistance decreases, and the augmentation index, which is a measure of arterial compliance or flexibility, improves. Prehypertension is a huge problem. These patients have pressures between 120 and 139. It's not treatable by standard pharmacotherapy, but within a year, typically 95% of prehypertensive patients are hypertensive because nobody wants to reduce their sodium intake, exercise, change their diet. So typically these patients become hypertensive. So we thought, can we reverse this trend? In this paper published several years ago, we took 37 prehypertensive patients and made 100% of them normotensive in 30 days, to the tune of a 12 millimeter reduction systolic, 6 millimeter reduction diastolic, 6-minute walk tests. The patients on the active were able to walk significantly further, suggesting an increase in functional capacity of the heart, and then a physical and mental composite score showed an improvement in quality of life. This is data published last year by Ed Lee, who's an endocrinologist in Orlando. He had patients that have had stable carotid plaque for the previous 3 years based on his therapeutic regimen. And so the only thing he did different was put them on Neo 40, 1 lozenge twice a day for 6 months, came back, remeasured their carotid intermediate thickness, and saw about 11% plaque regression. Now, if you look at the meta-analysis on aggressive, very aggressive statin therapy, getting cholesterol levels in the basement, you see about a 2.7% regression after 3 years with statins. We see 11% regression after 6 months. This is new data looking at 24-hour ambulatory blood pressures after a 60-day regimen of Neo 40. And what this shows is that after 60 days, you completely reset the rheostat on blood pressure. That steady state blood pressure becomes more normalized. So we're changing not only endothelial function, but the progression of changes in blood pressure and even the response to therapy. This is a patient with Raynaud's syndrome, cold hands, cold feet, microvascular disease. 10 minutes after the lozenge, open up the microcirculation, you start to perfuse the digits. And I'll end with just this, going back to this Nature Medicine paper we had in 2011, this inborn error in metabolism. In working in the Texas Medical Center, you get to collaborate with a lot of talented physicians. I'm a basic scientist. I don't deal in patient care, but what we do is we can take patients' problems, model them in the lab, and then go from bench to bedside, back to the, or bedside to bench, then back to bedside. And so there was a group of patients that were born with a condition called arginosuccinic acid urea. It's a urea cycle disorder. These patients are born without an enzyme called arginosuccinate lyase. So obviously it's a urea cycle enzyme disorder. They develop arginosuccinic acid. It's excreted in the urine, and that's the basis for the diagnosis. But as these patients are treated for the recurrent hyperammonemia with diet and drug therapy, they're still developing a lot of neurological dysfunction, progressive liver dysfunction, coagulopathies, resistant hypertension, and renal dysfunction. And so Brendan Lee, who was a Howard Hughes investigator, came to me in 2005 hoping to model this in the lab to try to figure out, help him better treat these patients. Because it appeared to him that it wasn't just a urea cycle disorder, there was something else happening. And so we were able to model this in mice and then take this back to the patient. And this is the lifelong blood pressure profile of this single patient. At 5 years old, he was diagnosed, had clinical hypertension, put on standard pharmacotherapy, and for 10 years he was really unmanaged. And in February of 2010, he presented to Texas Children's Hospital in the PICU. His blood pressure was 185 over 130. At that time, we knew it was a nitric oxide problem, so they dosed him with isosorbide, and he went home within 4 days, and he did quite well. But over time, standard with organic nitrate therapy developed tachyphylaxis. So he came back almost a year to the date, February 2011, taking 20 milligrams isosorbide 4 times a day, and his blood pressure was 185 over 115. We gave him a lozenge. Within 4 hours, his blood pressure was 135 over 80. We're now 5 years out, 6 years out. He's off all prescription medication. He graduated high school. This was on ABC Headline News. We published this back in the 2011 in the American Journal of Human Genetics. But to be able to overcome an inborn error in metabolism and to really correct a lot of the metabolic phenotypes of this rare genetic disorder simply by giving nitric oxide is really a feat. And these are the nice emails you get, and this really makes what I do worth it, because these are the nice emails you get at night from the parents. Not only have we basically saved him from his hypertensive crisis, but actually his cognition's improved. And he's able to beat his dad in chess for the first time. And I think I may show, I don't show it here, but he had severe left ventricular hypertrophy. A 15-year-old kid had the heart twice the size of a normal 15-year-old. Within 6 months, his heart had completely remodeled, left ventricular hypertrophy. All the dimensions of his heart had normalized. And so then this begs the question, I'll end with this because I think a fundamental question you have to ask yourself if you're intervening in patients, is this what nature intended? Are we tricking physiology, or are we restoring and recapitulating physiology? So the question we asked, and we didn't get these answers intentionally, these answers came to us indirectly, but is increasing nitric oxide production, plasma levels of nitrite plausible in humans, and is this a natural phenomenon? Through a collaboration with Case Western and an anthropologist who was interested in looking at the adaptive effects of people living in Tibet, which is 12,000 feet above sea level, partial pressure of oxygen is low, it's a very hypoxic environment, but yet these people seem to operate normally. They don't get altitude sickness, they don't get high altitude pulmonary edema. So how do these people adapt this low oxygen environment, where if we go to Tibet, we get acute mountain sickness? And so they had some studies showing that even though they're breathing in less oxygen, they're delivering more oxygen per unit time to the periphery. And so they came to us and go, how is this? How are these people regulating blood flow, delivering more oxygen per unit time, even though they're breathing in less oxygen? And the message is that because they're generating more nitric oxide. So this is the EPR signal of NO bound to hemoglobin in the Tibetans, you can see the nice EPR peak there. Plasma levels of nitrite and nitrate are about 50 to 100 times higher than people live in Cleveland or in Houston. So this is an adaptive effect. We've actually been able to recapitulate this. So if we take people that live in Ethiopia, which is about 6,000 feet above sea level, they have intermediate levels between us and Tibet. And if you take athletes and take them to altitude and train for a week, you can increase their nitric oxide output. So the performance-enhancing effects of training at altitude aren't just erythropoietin and hemoglobin production. It's because these athletes have increased their nitric oxide output. The problem is if you have endothelial dysfunction in older people who ascend to altitude that can't adapt because their enzyme can't make nitric oxide, those are the people who develop acute mountain sickness because they can't adapt to this low oxygen environment. Then that begs the question, is there the other end of the spectrum? Are there patients that are completely devoid of NO? And if so, what are the clinical consequences of this? And again, this wasn't a question that I deliberately asked. This answer came to me from a nephrology buddy of mine who's been hemodialyzing patients for 40 years. And even though he corrects their cholesterol levels, their blood pressure levels, their calcium levels, 70% 5-year mortality from cardiovascular-related accidents. So the question I asked was, what is hemodialysis doing to nitric oxide output? And this was just kind of the design of the study and kind of the historical aspects of these hemodialysis patients. So we took blood from the fistula of patients going on hemodialysis, analyzed it for nitrite, nitrate. We took blood coming directly from the hemodialysis machine after one cycle. Most of these patients dialyzed for 4 to 5 hours and then take from the fistula these new steady state levels. And hemodialysis removes about 85% of the plasma nitrite nitrate. We actually do endothelial function tests on these patients before and after hemodialysis. And even though you're removing liters of fluid from these patients, their endothelial function actually gets worse. And it's surprising to me because as we're in the dialysis center, these people are eating pimento cheese sandwiches and potato chips while they're getting dialyzed. And so their endothelial function's horrible, their diet's horrible. And if we collect their saliva before and after, the salivary concentration of nitrite nitrates are moving about 95%. So these people aren't getting enough nitrate from their diet. Their enzyme that makes nitric oxide is dysfunctional. And so no wonder they're dying from heart attacks, strokes, even though you're correcting all the other cardiovascular risk factors. Next is let's look to nature for what nature intended. My wife gave birth to a healthy baby boy about 9 years ago. We were leaving the hospital. It was my first kid. And so the discharge nurse goes, well, you should breastfeed, but if you can't, then give the baby the formula. And so the scientist in me came out and go, what's the difference in what we're given here? So after a brief struggle, I convinced my wife, when we left the hospital, to come to my lab and give me some of her breast milk to analyze it and compared it to the formula. And this is what we found, that the colostrum, this was day 2 postpartum, that the nitrite in the colostrum was actually much higher than any biological fluid would ever measure. And to rationalize this, I went and got an IRB from UT and Memorial Hermann Hospital, and so we began to see if this was just an aberrant property of my wife or if this was true throughout nursing mothers. And what we found was this pattern was apparent in, I think we had 71 patients. And so the colostrum, day 1 to day 4, was high in nitrite. And then as it matured to transition milk to mature milk, this ratio changed. And because babies are born sterile, they don't have these nitrate-reducing bacteria in their oral cavity or in their gut. So nature's developed a way to deliver nitrite instead of nitrate, to deliver nitrite to the baby until the gut flora and the oral microbiome begin to colonize. Then the breast milk can secrete nitrate and the babies can then utilize that. This is different in vaginally delivered babies versus cervical or cesarean delivered babies. The other thing that, and I don't show you this obviously because of time, but necrotizing enterocolitis, we've been able to model this in mice, that premature babies born under chronic hypoxia develop necrotizing enterocolitis, and you can completely overcome this and protect from NEC simply by giving nitrite in the formula of these mice. So I'll just conclude and let you eat, and I won't bother you anymore, but hopefully I've convinced you that nitric oxide is critically important, and your patients cannot heal no matter what chronic disease they have, without consideration of nitric oxide. And in my opinion, I think you cannot practice any type of anti-aging or restorative or integrative medicine without considering nitric oxide as kind of a first-line therapy. And I think what we've discovered here we're trained in kind of hypothesis-based research, but I think these are fundamental truths because this is what nature tells us. And when these fundamental truths pass through three stages, first it's ridiculed, it's violently opposed, and then it's accepted as being self-evident. I've experienced this in my own academic career. I've been ridiculed for 10 years telling people that you can affect blood pressure by changing the oral microbiome. I think now it's violently opposed. What we're doing is very disruptive to a lot of big pharmaceutical companies, to the standard of care. But I think these fundamental truths will eventually become self-evident because you cannot ignore the data at this point. I'll leave you with one good reference. This is a book I published or edited with Joe Lascalza, who's chair of medicine at Harvard Medical School and chief of cardiology at the Brigham. This is the second edition that just came out a couple of weeks ago. But we have 25 authors of this textbook that talk about nutritional epidemiology, cancer formation, exercise performance, hypertension, all included in this book. In fact, Lou Ignarro, one of the Nobel laureates, wrote the foreword of this book. And so it's a great reference, and with that, I'll leave you my contact information, and I'll be happy to answer any questions and allow you to eat your supper.